Hi, my name is Victor Sumskoy. Yesterday I made a presentation at BarCamp Sarasota on genome as a program, and many people asked me to record this presentation, and that's what I'm gonna do right now. I just have one favor to ask you. My name is Sumskoy, and I noticed that for Americans, three consonant letter in a row practically unpronounceable. So don't try to shorten it or change it somehow. This is not Suskoi, not Sumoski. And if you just cannot pronounce it, just say Victor. Maybe that's enough. Thank you. So, my name is Victor Sumskoy, and I call myself software enthusiast. And I have more than 40 years of experience in software development. And that's come to the question that I was a part of information technology revolution, which happens in the last 40, 50, maybe 60 years. And I was proud that I was not only witnessed this revolution, but I was a part of this revolution. But anyway, like any revolution, it can come to kind of a saturation and maybe slow down. And already 10, 15 years ago, I started to think about what can be the next big thing. Sure, I didn't predict that revolution is not finished yet, and we have right now mobile revolution. But anyway, the question was, what can be the next big thing after the information technology revolution? And what I came up with, I thought that it's gonna be a revolution in biology, and 21st century gonna be century of biology. And it's give me kind of a frustration because I cannot be part of this revolution. Not because I cannot learn new thing, but maybe just because I don't like biology and chemistry, to put it mildly. And this was kind of a frustration for me. And just a few short, maybe 5 weeks ago, I decided to look at biology and especially at genomics as from technology information point of view. The area I know a lot about, but it can be probably somehow applied to genomics. And that's what I'm starting from. So I start reading about DNA. And this was my first strike, I would say, for me, first discovery which I had. Because I found out for myself that genome in DNA, which I always thought this is a program, kind of a program, I never had a doubt about this. And it contains only 4 nucleotides. And they named like A, C, G, and T. It's their names. For me, it doesn't matter. From an informational point of view, it doesn't matter how they call. What is matter is there are only 4 of them. And that's what make me very anxious as IT professional. Because try to imagine, not 3, not 7, not 20, but 4. This is a perfect base for digital system, quaternary system. So I understand that I can digitize DNA in a normal hexadecimal form, where each letter will be presented with 2 bits. And if you are not very familiar with digital systems, the most used digital system is decimal system, where we have numbers from 0 to 9. We in computers use binary system, which has binaries and hexadecimals, but quaternary system, this is a part of such a system where we can have numbers from 0 to 3, and this is a perfect for digitizing. My only question was, what letter correspond to what number? And I made just one suggestion, and this suggestion is that nature is rational. And so I decided to arrange this letter, it's these nucleotides, but their weight. And that's what I come up with after all. This is how I can present any nucleotide in a binary form and put it in a normal binary representation. And that's what I did. And so I found some DNA sequences which are freely available from internet, and I found that they have lots of different formats. But for my purposes, for purposes of digitizing DNA, the most suitable format was a FASTA format, which has mostly just nucleotide sequence, probably with some comments. And that's what I did. Each letter in there I transform into 2 bits, and so every 4 letters will be 1 hex numbers. And so that's how this DNA can be presented in binary form, which I called hex. And so each number is like 98 in hexadecimal form. It's 2120 in quaternary form, which represents these 4 nucleotides. But, for example, I had 60 million numbers. I had 60 million letters, in fact. And now I have 15 million numbers. And I really didn't know what to do with them. What possible I can do with this? So I decided just to play with numbers. And probably I can find some fundamental mathematical constants in there. Constants. And what I did, I've heard something very suspicious about, or very interesting, I would say, about human Y chromosome, which, as I heard, not changing much for thousands and thousands of years. So I pick up this Y chromosome, human Y chromosome, transform it into hex format, and try to find, say, constant pi. I understand that because of I have binary form, they do not recognize decimals. So I found a hexadecimal presentation of this pi, and didn't find anything. So I thought maybe pi, this is not very related to life or to gene. So, but there is some more fundamental constant which can be related to a human, and this may be golden ratio. And I tried to find golden ratio without success. So I postponed this for a while. What I did next, I tried to figure out how I can visualize all these 15 million numbers, kind of a graph. And that's what I decided to do. I will try to take 2 sequential bytes and pretend that they're coordinates somewhere, x and y coordinates, on some particular surface. This gives me ability to put all these numbers in a perfect 256 by 256 matrix. And this particular, for example, 2 bytes will be a dot somewhere on this matrix. So far, so good. And so what I did, I take this Y chromosome, human Y chromosome, read first 64 kilobyte of hex data and put it on screen. And what I expected to see is just random dots somewhere. And then I start to read next 64K, next 64K, and show it on screen. And that's what I have. I read more and more and more, and I see that there is a definite pattern in there. That's a human matrix. I was shocked. I was really, really shocked. This is a human matrix. That's basis of our life. And it's even scary. And I can't imagine. Maybe this is sort of my presentation of my hex data, which I did. So first I try to do, I take another binary files, for example, zip file, to put it through the same procedure. And it looks pretty random to me because zip file by definition doesn't have any organization. And so I take executable. It has got some kind of a patterns, but it's very far from a human pattern which I saw. And the next was question: is this just for human pattern, or maybe this is for some another species too? And so I pick up mouse, this mouse chromosome 1, and I see absolutely the same pattern. And then I try lizard. It's not a mammal. It's a lizard. It's very far from mammals. And I see the same pattern. And I had tried to do this procedure for many other species like dogs and cow and fish and wild boar, orangutan, and they all have the same pattern. It was something. It looks like I discovered something which nobody saw before. Until I even try plants, and they not exactly, but patterns still recognizable. Until I find worm. That's a different pattern, definitely different pattern, because I have worm, I have mosquito, which for example this pattern resembles worm. This is worm, this is mosquito. And this is, for example, honeybee. They are, all three of them resemble each other, but very different from pattern I saw before. And then I try E. coli bacteria. It has another pattern, and this is influenza. This is kind of a cheating from my side. Influenza, that's a virus. And so as a virus, it has like just a few hundred of nucleotides. And so if I take just one virus, it would be just a few dots on this matrix. But this original file, which I use, has a size of 300 megabyte. And that means that something wrong. It's not one virus. And when I look at it inside, I saw that in this file they put all known different influenza viruses they are aware about. And some of all these viruses gave me very recognizable pattern. I don't know how to explain this, and I don't know what to do with this. But first I decided to give some kind of a definition to the matrix I just discovered. I called it SumMatrix because it represents frequency distribution of a sequence of 8 nucleotides. So each element of this matrix, for example, if we have a dot in here, 87F in hexadecimal, it can be represented in quaternary form like this, and it represents sequence of these 8 nucleotides. And that's why I called it SumMatrix. This is kind of a statistical signature of genome, how I can see it right now. And this presents, as I told you already, a frequency distribution of 8 letters. I called it codon, though it's maybe not right from genomic point of view. But this is codon, which I can see 8-letter codon. And the bigger value in the cell, the matrix, the darker it looks in the SumMatrix presentation. And that means the more often this combination occurs in a genome. And less occurred sequences must be more interesting because they represent this pattern. And I try to theorize what can be this different rare combination be. Maybe this is mutations for better organization. maybe this is really occurred markers which separate important chain of nucleotides. Maybe the pattern defines level of genome complexity, or maybe it's even a level of its perfection. And I really don't know what to do about this. And I understand that many investigation is required, and lots of now digital analyzing can be done as soon as we have any ideas what to explore. And I understand that SumMatrix, original SumMatrix, has absolute number in these counters, which represent each dot on this matrix. And so I come up with solution how to normalize such matrix so it would not depend on number of nucleotides in chromosome, for example, and this is particular chromosomes. And I come up with a solution which introduce 8 levels of normalization, with level 1 where I have only 0 and 1s instead of counters. And this gives me idea that such level 1 normalized SumMatrix can be easily compared with another SumMatrices of another species, or it's maybe much easier for future analysis. And now I have kind of a pause. I was so excited about it that I decided to make a movie, and I would like you to see this movie. This is cow chromosome 1. This is the cow chromosome 2. Can you see the difference? I can't. That's lizard, I told you. This is a primitive fish. That's a human. That's a viruses. Birds, fish. Pattern is very recognizable. This is east. Wow! I still can see kind of a pattern, maybe very different. Not very different, but there is a pattern over there. And now I see very different pattern, which is in worms and mosquitoes and honeybees. I think that insects has different pattern. Wow. That's a virus. That's what I expected to see. Just random dots. Wow, we are all alike. We are all alike.